Yes, so I will have a very reminiscent, but yet very different system I will tell you about and guide you on how fluid flows shape life. But to set the stage to check how we can understand the complexity that we see in living systems, let's check our own complexity ourselves. So here I have a task for you. So here I'm showing you a maze, and the red dots mark the two exits to the maze. So your task now is, what is the shortest path between the two red dots? Let me guide you a little bit in the back. This is the outline of the maze. Solve it. I bet you did, right? So this is the shortest path. Okay, well done, great. Okay, you're a multicellular, system, but would you bet that a single cell can do the same task? And this is what we're going to witness now. So what you see here, these yellow dots, this is Pfizer. These yellow dots will merge into a single cell, gigantic, and we're going to guide the single cell by marking the two exits with this beloved food source, oat flakes. And over time, this single cell reorganizes its morphology such that there's only a single connection left that's actually tracing out the shortest connection between the two exits. So a single cell can solve the maze. My task today is to convince you that we can aspire to achieve, to understand this complex behavior as a function of the fluid flows that are flowing through these tubular structures. So, because if we think about flows and we zoom into a single tube, then just by having flow, the flows provide information to the tube. Because just by having a flow, the flows provide a shear force that's acting on the tube wall, the flows exert a hydrostatic pressure, and the flows can transport signaling molecules. And now, in a setting where these are not the pipes of your apartment that are made of steel, but instead these tubular walls, they are active. So that means that they can respond to this information. And what does it mean? So let me zoom out into this network again. Say, in this whole network, we have a tube, and this tube gets signaling molecules that drive this tube to dilate. Now, what happens, because this tube got bigger, is that it's sucking in more fluid flow, which means that if this bigger tube is taking more flow, then also the entire rest of the flow in the network is going to be rerouted, which also means that signaling molecules that would be transported with the fluid flow, they would be routed to different parts of the network. And that means that also other tubes now may respond to signaling molecules that all of a sudden they do get. And you can see how a feedback between flows and network morphology unfolds just by this one idea that the flows just being there provide information. And so I want to understand the feedback between flow and morphology. And the general realm of here we are, this was a biologist, this is a physicist. We're going to look into one model system which has all the ingredients for me to understand what I'm coarse graining down here to flow and network state. In fact, I have a little bit of a guidebook of what I'm looking at, because I want to understand what is actually the local information that the tube is reading out from the flow. I want to understand what is the role of the network topology in shuffling this information around. I will tell you how the network hierarchy, so the pattern of where I have thick tubes and thin tubes in this network, actually forms a memory of information provided to the network. and then at the very end guide you a little bit of how I believe we can take what we learn here to really control network functions and the various plethoras of flow networks that we have. But before I dig in, let me tell you a little bit about the system that you saw solving the maze. So what I have here is Physarum polycephalum, which is a slime mold. And so, big disclaimer here, a slime mold, yes, it sounds like we're talking about a fungi because of the term mold. And the reason why this is called slime mold is because, I mean, it should remind you of the networks that we just saw in Toby's beautiful talk, right? Because you have a network structure that is crazily reorganizing over time, yet and again. And that's why it's called a mold, because of this apparent visual similarity, but it's not a mold. It's not a fungus. So really, if you look at the genetics of slime molds, they are halfway half fungi and half animal. And you even have, a bunch of very good, sources that, plants have as well, right? So it's genetically very, very distinct from fungus. And you will see so when I'm going to talk about how this network is actually set up. Let me, just for the fun of it, actually play this one more time. So what we see here is actually, this is an arena of food sources, and this is a single cell. It's truly a single cell in the sense that it's set up as a syncytium, so there's no cell walls that are even broken down. There were no cell walls from the start with. So it's really this tubular structure that can grow to big sizes. This is like a centimeter. You can grow it to square meters. And it has this, it just happens to be shaped into a network structure. And it has thousands and thousands of nuclei within the structure. Now, if we zoom into this network, we see very organized fluid flows that are here sped up in time, and you see that these flows are flowing in one direction and they flow back in the other direction. And so what is going on here is that these tubes are built out, as I said, as a single cell, right? So you really have, like in an animal cell, so these cells are lined with an actin cortex, and the actin cortex is enclosing the liquid cytoplasm that then holds all the organelles and so on. Now here, the actin cortex on the tube wall is undergoing rhythmic contractions. And so when a tube segment is contracting, it displaces the cytoplasm that it encloses, and then the cytoplasm starts flowing. And so these contractions, just to give you a time scale, so that's typically on a 2 minutes, every 2 minutes they contract. And now I want to, and these are really happy Poiseuille flow. So if your mind is set on, flows in your piping system, except that, they're driven by contractions rather than a pressure gradient, you have the physics perfectly right. So this is simple laminar flows, by no means as complicated as in the fungi networks. So, but these flows, as I aim to convince you, are very important for the system. And just to give you a taste, these contractions that are driving the fluid flows are actually coordinated across the entire network. So this is mapping out the phase of the contractions for you to see that there's a traveling wave that's sweeping this entire network. And that gives rise to really a traveling wave that's driving the fluid flow throughout the network, swishing forth and back, thereby creating really the strongest flows that you could achieve by the mechanism of local pumping across this entire network. Another way of how we can see that really the system desperately cares about having strong flows and thereby also good capabilities to transport resources is to poke it. So say we take a network and we put down aversive blue light in the center. it rapidly makes sure to reorganize its body to go away from this blue region. And it does so, again, by subtly reorganizing flows. And just to appreciate what is going on. So we have this contraction. Think about that the contractions of a tube, it's oscillating in a cosine manner. But what you actually, in fact, have is you have a dominant mode, and you also have, say, you have an overtone of like twice the frequency, here mapped out in yellow, and the higher frequency in orange. And now, what the organism does when challenged with this blue light, I really want to get away from this, it modulates a very subtle parameter, namely the phase difference between the overtone and the dominant mode. And what this does is pictured up here. So when I shift like the phase difference between yellow and red, green is showing the sum of both of them. What I do is I modulate the occlusion of like how tightly a tube contracts. And obviously, like if I take a tube and I contract like that, I generate less flow. If I contract tighter, I generate higher flow. The reason why I'm showing this is A, to emphasize the system is organizing response by modulating flows, and it does so with an active mechanics approach of changing dynamics in the active cytoskeleton that's driving these contractions. So these flows are important. If we now go back to the setting of like how can it solve the maze, the question is how is information transported? And so we tested that by like taking a network and putting down a droplet of food. And so if you do that, like the literature said, like if you put down food, it increases the amplitude of these contractions. Now, there's food actually has to propagate into the network. So we should see a propagation of this increase in amplitude. And this is what you see here in this camograph. So the camograph is tracing out this magenta tube here that's blocked by the magenta line up here, and over time. So you see here now dark, bright, these are the contractions of this tube. And this is the region where the droplet hit in the center. And you can here now nicely see how the amplitude, the difference between bright and dark, really ramps up, and this really propagates into the network. And so the important thing is it propagates with the speed of transport with the fluid flow. So no wonder the system cares so much about the flow. The flows are the means to transport information. So, but it's only good to transport information if you can actually make use of that information. And so to make decisions, you need to have a mean to store information. And so this is the story that I want to dig in. So I'm going to show you this movie again. You saw it. You get another task. So your task, you're familiar with this, right? These were the food sources. And this will migrate around. So your task, you will follow the center food source. And we play the movie again. You see that the food sources are entirely eating up. There's no trace left of the food source. Oh, sorry, I didn't play. There we go. It's entirely eaten up. There's no trace left of the food source when it's eaten. Can you tell me where the center food source was? I bet you can. Why can you do that? Because there's this thick ring of tubes that is encircling where the food source was. And doing this was a Friday afternoon experiments. Like this is a Friday, like I should really emphasize, you should do Friday afternoon experiments. Seriously, this is a Friday afternoon experiment that, okay, honestly, it became a weekend experiment because that was very challenging. It's a data set that Mona took, PhD student in the group, because we didn't have a top stage incubator at the time. So it was very challenging to have data of looking at Physarum crawling around for days. And so she actually slept next to the microscope and she had set up a woolen string to a reservoir of water, and she was using the capillary forces to make sure that the petri dish with the agar and the slime mold on it would get the humidity through that woolen string to keep it from drying out so that she could image it for 3 days. But it really was, in mindset, it was a Friday afternoon experiment. This is a 4-year-old data, 4-year-old data, 5-year-old data, and we're still looking at it on, I would say, a weekly to monthly basis. So why was it so inspirational? It triggered the idea that, wow, like, you know, where I have thick tubes and thin tubes is a memory. So, you know, is this really a memory? This observation started to dig in. And so there's 2 questions, like, you know, how would it work to encode the position of a food source into the network architecture? And is this really memory? And now the real work starts. This is the fun part, the Friday afternoon. So the real work was then to do lots of data sets so that eventually we had this prime data set. What this prime data set has is that the network is actually stable and not migrating away, so that we can really quantify the dynamics going on in the network. And actually, I should emphasize, so the joy of working with Physarum is that it's inherently a planar network, so every connections that you see, these are actually like, you know, actual connections compared to fungi, where you have no idea if they're like just happen to lie on top of each other or if they're fused. And I should also say that by now, we can not only have the total view of the network, but also the flows throughout the network. So we really have access to all physical parameters, which makes it a wonderful system. So here, we're doing the same experiment. We're putting down a food source, and we're looking at how does it change the network architecture. But it's much more controlled because it doesn't migrate away. So we put down the food source at 0, and we see that within 15 minutes, there is the tubes very close to the food stimulus. They dilated, but also further back, a new pattern of very thick and thinner tubes nearby was imprinted. And it stays. So this pattern that was imprinted does not change over the next hour. So how does this change in network diameter come about? So what we do is we now look at the average tube diameter. And like tube diameter now, I should emphasize, like I know that the tubes are contracting all the time. I'm just taking, you know, the base diameter, sort of averaging out these fast contractions. And so now we're looking at the tube diameter, and we're ordering all these tube segments throughout the network from close to the food to further away. And we're normalizing it by the diameter that it had before at the white line we put down the food stimulus. And then we see that there is a wave of dilation traveling in from the food source into the network. And it also goes hand in hand with the rest of the network shrinking. I mean, it's a finite mass, right? So if I grow some places, others have to shrink. Now, based on that, this created 2 hypotheses. Namely, if you put down a food stimulus, there's rapid dilation, so there's some softening of the tube walls that's going about. It happens within 50 minutes. This is the only thing that makes sense on these timescales. And we see this wave of dilation. It again travels with the speed of how particles would be transported. And so this softening agent has to be transported with the fluid flows. Now, having this hypothesis, we turn to theoretical description to make predictions that we can then verify in experiments. And so to achieve a nice theoretical model, we did one more step of simplification, which is to note that in the end, these dynamics of this softening agent spreading into the network is actually a function only of the distance to the food source. So we can actually describe it as the spreading of a cloud of concentration also in a single tube. Now, this is important because now we can really describe the entire system. We know the physiological parameters, so we can really nicely describe the flows that are arising due to contractions, And so the only hypothesis that we put into the model is that we put down a softening agent, and as it travels, it softens the tubes. And so now if we look at these dynamics, we observe the following, that tubes close to the stimulus site, they, after the stimulus, again at time 0, they rapidly dilate. Tubes further away will first shrink, conservation of fluid volume, and only later on, when the softening agent actually also spreads to the screen part, they will start to dilate, which gives us access to another quantifiable parameter, namely the response time until this segment further away will actually start dilating. And so now, having this from the model in mind, we go back and compare to our experimental data. And so what we now observe is that if we take all the tube dynamics and we automatically classify, are you straight dilating or are you first shrinking and then dilating, we observe the same characteristics. Close by, just dilation, further away, first shrinking and then dilating. And even the scaling of this response time when they start dilating is scaling correctly as predicted with the parameters in the system. So this drives us to conclude that indeed you set up memory by the localized food source driving the release of a softening agent within the network, and it's transported within the fluid flows and sets up a new hierarchy of thick tubes and thin tubes. Now, is this really memory? So, okay, memory is a not well-defined physical term, so I'm going to define memory by saying that if it's really memory, then I should see that it's being read out in the response, the behavior of the organism. And so now the challenge is I can't recreate exactly that same network and not put the food source up there and instead put it there, because there's no such thing as two identical individuals. So, but what I can do is realize that actually the network that I started with did have a hierarchy, right? It did have thick tubes and thin tubes to begin with. And where these thick tubes were impacted the response to this food stimulus, as we're going to just look at this data set one more time, because you see that these thick tubes that were oriented to where we put down the food source, they get really enlarged, whereas the thick tubes, that was actually much thicker down here, that was further away and oriented in the wrong direction, it actually shrank over time. So where you have thick tubes is get read out in the response to new food sources. So it does constitute memory. And just to highlight, actually going back in time to a previous project, the hierarchy does impact transport. So let me, just to drive this home, show you the following simulation by Sophie. This is an actual network morphology of Physarum, and what we do is like these two simulations, they have the same mass, and we're going to look at the transport of particles, but here we made all tubes to have the same radius, and here we have the actual radii that they have. So if I follow the dynamics, I see in the uniform radius, I see the following spread. Now if we embrace the hierarchy that the actual network has, we observe that there's a much faster and also much more oriented response transport within the network. So really the pattern of thick and thin tubes matters. So memory. We find that there is memory in the network architecture. Let's do a thought experiment. Remember the challenge from slide 1? How about now we put down not one food source, but two food sources? And okay, so let's happen to take this data set where we constrain the network to be confined into this weird shape. So what is going on now? So we have now these two food sources that are dilating the tubes, like they're releasing a softening agent close by when we put the food sources, and these softening agents get transported into the network. So where do they go? Well, they go to the tubes that are closest and best connected, right? So obviously, those that are closest to the food sources, they will keep growing at the expense of the rest shrinking. So indeed, this is how just by having an active response of the tube due to a softening agent, you can solve a maze. So this is to show you that we here studied an impact of how signals that are transported with the flow provide the local control that is being read out by a tube wall, and that the network hierarchy, so the pattern of thick and thin tubes, provides memory of the past, and if you, with the past, can impact the future, it has this amazing perspective that you can actually program a network. Just to say that this is obviously just a snapshot of a lot of different works that I don't have time to go into, but just to put it up. And I would actually like to just show how working on Phasarum for me is a very accessible flow network, but it's really embedded in this much richer and bigger realm of flow networks in general. And also just to say, I really enjoyed this meeting, and just talking to Massimo yesterday. Obviously the next step is now to think about how if you store memory, how can such a network learn when you think about Phasarum. But actually, let me take a few minutes to tell you of how I believe what we learn in the physical context of Phasarum, of how flows and morphology feed back, is inspiring to think about, having chemical or biochemical control of network function. And so, because, flow networks are everywhere, and a prominent example is actually, porous media, which is the rocks or the filtration, your battery, fuel cells, they all rely on that you're pumping flow through a random structure that in the end, this is a tubular system of pipes with large flows and low flows. And here really the network structure is really highly impacting of how this has more disorder, this has less disorder, of how also molecules are transported throughout these systems, which is very important if you think about your battery and you want to get the ions across it, right? And it's very inspiring now, having this theoretical insight on flows and transport in these man-made static networks, and seeing that actually, if we go to this high disorder, we're actually look that the flow is actually just channeling through this single route, and there's all these large regions. There's not much flow velocity at all, right? Can we hijack the methods from nature and make them better, make them self-organized to have first a better morphology, but also maybe teach them to memorize, teach them to learn, building up these systems. And so just to show a glimpse of what we recently achieved in being able to do is that we can actually build microfluidic designs that are built of hydrogel structures, and now these hydrogels we can drive by enzymes. And this is just a simple experiment of these hydrogel structures, and there's an enzyme that we put in and we drive them to deswell. And so that's the first step to now actually put in more complexity in the system. So this is one direction, looking into taking the physics that we learn and building them up in a lab, which helps me as a physicist to also understand these feedbacks. But another direction is also to say salmons are not ecological or medically relevant, but in fact, our vasculature is also a flow network, and that is of high medical relevance. And so there's also a different strategy in the lab, spearheaded by Fatimi, which I can't eye in the audience, but she's there she is, where we're now growing a vasculature on a chip, and we hope to really, with the same spiel that I showed you with Phasarum, assess how these vessels that are forming here, and we can perfuse by these inlets and, you know, look at the flow and how the morphology is evolving over time, really aspire to learn how we can program a vasculature, our human vasculature. So with that, I would like to thank you as an audience and highlight that I believe the physics of flow networks is a very rich field that we are combating by combining experiment, quantifying, and theory on various systems. I wouldn't be here without funding, but most importantly, without wonderful people. And just to flesh out that this is just a glimpse of the flow networks and the dynamics that we should care about. This is us. This is just a short list of the flow networks that we have in our body. And there's so many man-made systems that we should care about as well. Thank you.
Right.